In the third and final module, we're going to identify some differentially methylated regions using whole genome bisulfite sequencing data from 3 matched tumor and normal colon cancer samples, so 3 samples in each condition. Unlike the RRBS example above, we have dense data now, and the smoothing approach is going to perform really well here, and we're going to see some really fun results. So we're first going to read in some data that's already been smoothed for us. This is part of the BS-seq package. We're also reading in, we're reading in BS Cancer XFit, and we're reading in something called keeploci.x. We'll examine both of these objects once we've read them in. And we'll run this line as well, which makes any necessary updates. First, we can use the help menu. BS Cancer XFit has its own help menu that will tell us a little bit about it and where it came from. And so here it actually has a reference to the paper that it came from. So we have this paper over here, and I'll show you a little bit from it later, but this is the paper where the data came from. And it's worth reading if you're interested in this, in epigenetics and or cancer. It's a great paper. So the data are the 3 tumor and 3 normal samples from this paper. And there's this other data object in the environment that we're going to look at. And it was called, actually it doesn't have its own help menu, but we can inspect it. So this doesn't have a dimension, which means it's probably not a matrix, but it's a vector. Sometimes you find that out the hard way, which I just did. Okay, so this is a vector. It's got length 597, 371. And before we look at it more, let's look at the BS Cancer XFit data a little bit more. Let's actually just ask R to show it to us. And because it's a BS-seq data object, it gives us the usual information. It tells us that we have close to 1 million methylation loci in the 6 samples, and these are the 3 tumor and 3 normal. This one has already been smoothed for us. It saves us the time of smoothing a slightly larger data object. And it looks like they smoothed it using the defaults. Going back to the keeploci.x, it doesn't have the 958,000 entries in it, so it doesn't seem to be the same size as the BSmooth, as the BS Cancer XFit object. If we inspect it a little bit more, we can just look at the first few elements. And this is a bunch of numbers. And what it turns out to be is it's actually telling us which elements of which CPG sites in the Cancer XFit data have passed their QC. And I previously decided to inspect the ones that are not in this list, the ones that did not pass QC, and they tended to be ones with lower coverage. And so they've already done some QC, and they're telling you which ones are of high quality and should be taken forward in the experiment. And you'll see that the next step down here, we're going to run the BSmooth t-test on the BS Cancer XFit data, but we're going to subset it to just the loci that have passed their QC. And so that's what this is. We're giving it the usual 2 groups, the three cancer, C1, C2, and C3, and the 3 normal, N1, N2, and N3. And now we're telling it to estimate the variance based on group 2, not on everyone. These are tumor samples, which are known to be highly variable. Every tumor is different. Whereas among the normal samples, the variance should be more within reasonable bounds. And so for cancer studies, when we're looking at differential methylation in cancer, it's common to base the variance on group 2. And so that's what all this is. We can run this command to run the t-tests. And then we can, and it'll run pretty quickly, we can inspect and plot the results. So it's a BS-seq data object. It ran it on just the loci that were in the keeploci.x list. On line 185, we can run the function getStats to extract the t-statistics from this analysis. And we can inspect what we got. And again, this looks like what we saw earlier. So in module 2, so we have our t-statistics, both the uncorrected and the corrected. And a little bit more about what this is. I showed you in module 2 that they seem to give very different results and have different variants. The paper that I showed you just a few minutes ago identifies that there are these vast hypomethylated blocks in cancer, and this is now well known. Let's see, we can scroll through this and I can kind of show you what we're talking about here. So here's a BSmooth plot, and this is actually by the same group who created BSmooth. Here's a BSmooth plot showing that the cancer sample, here's a vast hypomethylated block, like the ones I was talking about. These are frequent throughout the genome in cancer when you compare tumor to normal. And they're very interesting, but after Hansen et al. Found these, they thought it would also be worth trying to find things like this. Like this is probably a promoter region. There are smaller differentially methylated regions where interesting things are happening that may be different than the hypomethylation that we kind of see on a broader scale. And so they developed the corrected T statistic in order to correct for this hypomethylated background in cancer studies. And so the corrected T statistic is used to kind of find these smaller DMRs even in the presence of these larger hypomethylated blocks. And so that's why we've been seeing these two types of T statistics all along. And so going back to our R analysis, we can use the DMR Finder function to find DMRs from this large set of T statistics that we have here. And here we're giving it, I gave it a pretty liberal cutoff in Module 2. Here we're giving it a very conservative cutoff. We're only asking to look at the most extreme test statistics. And the reason I'm doing that is actually so we'll get just a small number of the most interesting DMRs, because we're going to make some plots and we want to look at kind of the largest, most striking ones. If we made this more reasonable, we would find a lot more DMRs, even though, as I think I didn't mention at the beginning, but this is just chromosome 21 and 22 that we have in this data set. Okay, so we can ask it to, well, first let's inspect our DMRs that we just created. And so it looks like it found 11 DMRs, even at this very conservative, stringent threshold. And so 11 is small enough that we can just look at the whole matrix of results. And so here they are all hypermethylated, which is interesting. They're hypermethylated in cancer. So the group 1 mean refers to the 3 tumor samples. They all look to be relatively hemimethylated, while the group 2 is close to zero methylation, so they're unmethylated. And we'll see this more when we look at plots, but it's interesting that they're all in the same direction. So here we'll use the plot many regions function to look at some B-smooth style plots for all of our 11 DMRs. And we need to run lines 189 through 192. There we go. Okay, so hitting return to see the plot. So this looks much better than what we saw with our RBS data, much better. So what we have here, if you look at the gray lines here, this shows that these are some very CpG dense regions. There's still some holes in between where there aren't CpGs, but now because we're doing whole genome bisulfite sequencing and we're trying to cover all of the genome, we don't see this sparseness that we saw with RBS. We also note that this CpG-rich region is consistent with being a promoter region. It probably is. That's where we see the CpG islands. And so what we have is definitely a CpG island. We can see that on the CpG island, the normal samples tend to be completely unmethylated, so the smooth lines are down here, and even the highest data points for these samples don't go very high. The smooth lines for the tumor samples go up into the hemimethylated region, and the points are kind of all over the place. The curves are not as consistent with each other in the tumors, which is the higher variance that I mentioned earlier. But this is really a beautiful plot. It kind of shows exactly what is happening in this promoter region in cancer when comparing the tumor versus normal samples. And if you want to, you can go through and look at all of the plots, and every single one of them looks equally beautiful. They all look like this. And there's another one. Here we have hemimethylation or hypermethylation in the cancer samples within the promoter region, and then outside it, methylation is a little bit lower, and this is consistent with that hypermethylated block. And again, this analysis is based on using those corrected T statistics. That is the default for the DMR Finder. If you want to do some work on your own, you can go in and change the defaults. I'll show you how to do that in a minute. And you could remake the plots for the DMRs you find that way, and then you'll see things that look a little more like hypomethylated blocks. But here what we're seeing is a lot of promoter regions that are hypermethylated in cancer. And so here's another one of them. And this one looks a lot like the previous one. And they all look pretty similar, so I won't go through all of them. I'll leave you to do that if you want to. Here's another one. Yeah, they all just look great. Okay, so I'm just going to skip through the rest, but you're welcome to go through on your own and look at these. And I mentioned that if you want to, you can run DMR Finder again with a different T statistic, and to do that I'll show you how on the help menu. Here you can see this is the help menu. Here are all the arguments. You can see there's this option called stat, and the default for stat is tstat.corrected. And we didn't specify that, so we just used the default. But if you want to go back in and rerun it for the regular tstat so you can see some of the hypomethylated blocks, you can go through and specify stat equals tstat in quotes. So I'll define something called DMRs2, DMR Finder, And I'm going to give it a different cutoff because when I use 12, we don't find anything. But when we use 10, we find some. So I'll go ahead and run this. And then I'm just going to change the code up here temporarily so it reads the DMR's2 object. And then I'll just run this code to make some new plots. Okay, and the important thing is that I need to actually change the DMR Finder and add the new argument. Going back to the help menu, we see that the argument should be stat equals tstat in quotes. Okay, so now I'm just going to run this whole little block of code here where I've defined something called DMRs2, which uses the uncorrected tstat. We will run this and we will start plotting. So the other thing we need to do is to have changed the cutoff. When we did it with 12, we actually didn't find anything. When we do it with 10, the DMRs using the uncorrected t-statistic are a little bit less extreme, but they're still interesting. So I'm going to run this code from lines 188 to 192. Okay, so hit return to see the plot. Okay, so this looks a little bit different than what we've been seeing before. Now we're tending to find these bigger regions where there's a lot of hypomethylation. The corrected t-stats were designed to edit that out. And this one looks really different, actually. This it still looks like CPG Islands, but it looks pretty different. Okay, so a useful conclusion to what we've learned in Module 3 so far versus Module 2 is that when we have dense whole genome data, the differentially methylated region approaches make the most sense. So we're next going to we can also use the DSS to detect differentially methylated regions. It does it in a slightly different way, but the results will be similar. And so we're going to start by using the same data. We'll just create a little data object that's the QC'd version of the data. On line 208, we can run our DML test. And the way DSS identifies the differentially methylated regions is we first identify DMLs, and then from there we'll find clusters of DMLs that are consistently differentially methylated and use that to identify differentially methylated regions. And so here we'll run DML test on the 3 tumor and the 3 normal samples. Okay, so now the run is complete. We'll use head to inspect DML test since it's going to be large. And this looks similar to what we looked before. This is actually going to be for all, I think there were 600,000 CPGs that went into this. That's why it was so large. Okay, so yeah, we tested for differentially methylated loci among all 589,701 CPG sites. We can now use the call DMR function within DSS to use this output to identify differentially methylated regions that are differentially methylated in cancer. And so again, we're using the same default values. We're looking for a difference in the 2 μ's of 0.1, and we're looking for a p-value threshold of 10 to the negative 5th. And these can definitely be changed. But for these purposes, we'll run the call DMR function and inspect the resulting object. And this ran pretty quickly, and it gave us 5 DMRs. And if we look at them, they are similar to the DMRs we saw when we were working with B-Smooth. We have hemimethylation in the tumor samples and no methylation or close to 0 methylation in the normal samples. Just like with B-Smooth, we can make plots. These plots are a little bit different, but I like the way they present the information as well. So the show one DMR function will make a plot. And I like these because they show each of the samples on its own plot. And this is good for having, when you have small numbers of samples, this is a nice way to look at it. So this is a promoter region, it looks like, and you can see that similar to what we saw with B-Smooth, we have a lot of methylation, and kind of on average we have hemimethylation in the 3 cancer samples throughout the promoter region, whereas there's very little methylation in any of the normal samples. The promoter region is left unmethylated, as it should be if we want the gene to be free to be expressed. And so this is consistent with what we've been seeing with these colon cancer samples. Another capability that DSS has is it's based on using replicates to estimate the dispersion. It's typical to have replicates in experiments, but even if you don't have a replicate, DSS has the ability to estimate dispersion through smoothing. By borrowing information from the nearby CPG sites, we can get reasonable estimates of dispersion, and so you can still carry out the study even if you have just one cancer and one normal. So in this last section, we're going to pick just one cancer sample and one normal sample. That's what here we're subsetting and creating a data object called bs.1rep. We will then run DML test again to call the differentially methylated loci first, or to perform tests for differential methylation of each locus. And so we'll run that. Okay, now the DML analysis is done. We can use call DMR again to identify the DMRs from the DML results. We can inspect the results that we got. And it looks like five differentially methylated regions were identified. One might wonder, are they the same as the ones we just identified using all 3 reps? They might be the same, they might be different. So this top one here is on chromosome 22. It started at position 195, 10987, ends at 195, 12084. And so that's not the top one. In the previous one, there is one kind of in the vicinity of it, 1911. It seems like we found one in the same vicinity, but the differentially methylated regions are a little bit different. We can do the same thing that we did above. We can try plotting one of the regions. We'll plot this one. And now we have just the 2 samples that it was based on. But again, this looks like the classic example where the promoter region is hypermethylated in the cancer samples and not in the normal samples. And so this ends Module 3.